Okay, let's go ahead and quickly go through the review. There's 3 parts really. There's part 1, and then there's the addendum, the little like added scan sheet that prints differently for everyone for some reason. It's the same file, but on different computers it downloads differently. So we'll go through that. It's just cardiovascular regulation, and then we'll go through part 2, which is on lymphatic. Okay, so hopefully you guys have already filled yours out so that we can go through this nice and quickly, so that those of you who have a practical today can go get studying, because I know that's what you want to do. So for vessels, know the progression of vessels through the systemic circuit. Okay, so when we first leave the heart, what do we go into? Elastic arteries, then muscular arteries, then, and arterioles go into capillaries. Capillaries go into, venules go into, what kind of veins? Medium veins, and then we go to large veins, and then the heart. Okay, so the progression of vessels. We talked about which layer is which in the blood vessel. So the tunica intima is the inner layer, the tunica media is the middle layer, and the tunica externa is the outer or external layer. And again, we're talking about the blood vessel wall. So if I section the blood vessel and looked at the actual wall, there are 3 layers when we look at arteries and veins. Where is the internal elastic membrane located? So right off the bat, what kind of vessels are we talking about for both of these elastic membranes? Arteries. So if I say anything else, it is wrong, right? It's only in arteries, not veins, not capillaries. So in arteries, so the internal elastic membrane is between what other layers? Tunica interna or intima, and the tunica media. Right, so between the inner layers, right? You've got your inner layer, your tunica intima, and then you have the internal elastic membrane, and then you have the tunica media. Right, so between the first 2 layers, in an artery only. Where's the external elastic membrane? I have no idea what that was. Excellent. Between the tunica media and the tunica externa. So between the middle and outer layer, right? In arteries. So between tunica media and tunica externa, right? So if you have like your tunica intima, your tunica media, your tunica externa, right? Those are your 3 layers. Your internal elastic membrane is between the first 2, right? Your external elastic membrane is between the outer 2. In arteries only. If smooth muscles, well, where are most smooth muscle cells? Tunica media, right? So the tunica media, the middle layer is muscle. If smooth muscle cells in the tunica media of an artery relax, what happens to the lumen? It's bigger. What do we call that? Bigger. Okay, so it dilates. If the smooth muscle cells contract, what happens to the lumen? It gets smaller. What do we call that? Constriction. Know the difference between arteries and veins when viewing vessels under the microscope after sectioning. So we have the couple of slides that have the pictures, right? And we just talked a lot about about those pictures. So we looked at like if an artery, if we section the artery, and remember that was like this really kind of tight picture. The lumen got really small like that, right? And then we saw this really thick wall like this around a really tight lumen. And remember when we looked at a vein, it was kind of like the lumen was big and it lost its shape a little bit like this. So that was kind of what the vein looked like. So arteries and veins. The vein appeared larger, right? What? The vein?
Like the lumen was bigger?
The lumen was bigger, yes, but not the wall. Right, so larger lumen, that's what I mean by larger. So larger lumen. The artery has smaller lumen because what happens to the artery when you take the blood out of it? What does it do? It constricts, right? Because remember, it's so elastic. So it constricts without blood. Just like a rubber band, right? If I'm forcing a rubber band out, the minute I stop forcing it, it shrinks, right? So your elastic arteries your arteries are super elastic. You take the blood out of them and they go, and they shrink up on you. So the lumen looks really small and the wall is thicker. What else do we see in an artery? Which layer is the thickest in an artery? The tunica media is thickest, the thickest layer. Which layer is thickest in the vein? The tunica externa is thickest. Which one do you see elastic membranes in? So arteries have elastic membranes, the veins don't. And then we said one thing about the endothelium. What is the endothelium, by the way? Simple squamous cells, right? But where is it? The inside, right? When we look at the tunica intima, essentially that's made up of a really thin membrane that we call the endothelium. When we look at arteries and veins, the endothelium looks different when we section them, right? What's the difference? In arteries, it looks all bumpy and pleated, right? Remember, because there was this nice, so if the artery started out with a lumen like that, and you had this nice smooth membrane, now if I squish it, right, it constricts and it gets really small. I've squished that membrane, so it folds. So you have a pleated endothelium like this because it gets crammed into a small space. The vein does not constrict. It kind of loses shape a little bit, but it doesn't constrict. It's not as elastic. So the endothelium still appears smooth. Okay, so arteries, the endothelium appears pleated. Pleated endothelium. That's not the case in veins. Okay. Cells of the tunica media and tunica externa receive nutrients and oxygen from what? Vasa, vasorum. Vasorum. Remember, that is like vessel of vessels, right? The vessels that feed the vessels. Because when you look at your vessel wall, you've got all these cells out here that make up the tunica media and the tunica externa, and those are living cells. They need a blood supply, right? They need oxygen and glucose and all that good stuff. And that cannot diffuse from in the blood because the wall is too thick. So you've got tiny vessels that actually deliver the blood to the cells of vessels. Okay, so what is an aneurysm? A bulge in the artery wall. Why? Why does it bulge? What's the problem with? The elastic fibers. Good. So a weak spot in elastic fibers allows the vessel wall to bulge, right? Typically an artery. So the artery wall to bulge out. They're supposed to be nice and elastic, right? So when there's a bunch of pressure pushing out on your artery, it just stretches. When the pressure goes down, it goes back to normal. That's it. When the elastic fiber's not working appropriately and that pressure pushes out, instead you get like a little bulge, a little bubble that starts pushing out on that artery wall. And that's weak, right? It weakens and it gets weaker and weaker and weaker. And it can burst. It can rupture the aneurysm. So what happens when the aneurysm ruptures? Okay, so if it's in the brain, we call that a stroke, a hemorrhagic stroke, not a thrombotic stroke, hemorrhagic, because it's a bleed. But what happens just in general? Like, just tell me what's going to happen if you've got an aneurysm that bursts. Good. Internal bleeding, and you interrupt blood flow. right? You can't deliver blood any longer because instead of going through the vessels to where its target is, the blood's all leaking out through that ruptured aneurysm. So you have internal bleeding and interrupted blood flow. Where are the two most dangerous spots to have a ruptured aneurysm? Brain and where else? Aorta, right? Because there's a ton of pressure pushing a ton of blood, so you bleed a lot and you can't deliver blood anywhere. Okay, so capillaries. We have multiple types of capillaries, right? Continuous capillaries, fenestrated capillaries, sinusoids. This type of capillary has pores in its endothelial lining. Fenestrated. And because it's got those pores present, it allows things to enter and leave the blood a little bit easier, a little bit faster, and then it also allows some extra stuff to enter and leave. So what does a fenestrated capillary allow to enter and leave the bloodstream? Okay, so water, ions, glucose, all of that stuff, it goes really fast, right? Rapidly. But then what extra stuff does it allow to enter and leave? Larger solutes, right? So amino acids, some hormones, nutrients. Of course the gases too, right? The gases can go in and out of anything. But this stuff up here is essentially the stuff that can go in and out of any capillary, right? Water, glucose, fatty acids, respiratory gases, oxygen, CO2, that can cross any capillary. But because the fenestrated capillaries have these nice sized pores, that stuff can rush in and out really fast. That's really good at exchanging stuff rapidly. But because these pores are present, we can also exchange kind of larger things. Some relatively big amino acids, even polypeptides, where we've got a couple amino acids joined together, hormones, some of our protein hormones, some kind of large macronutrients. So why is that important? Where would these be important? The kidneys, where we filter out big stuff from the blood. Where else? Intestines, right? And the intestines, that's where we're absorbing tons of nutrients. So we need to absorb nutrients really fast, and we need to absorb like big macronutrients. So we need these fenestrated capillaries to do that. And then because of hormones in some endocrine glands where we make those hormones. This type of capillary allows for the exchange of very big substances like plasma proteins and blood cells. Sinusoids. Remember they've got, okay, so actually here, why can these large things enter and leave? What do sinusoids have? They've got large gaps between endothelial cells. Okay, so the fenestrated capillaries have little pores present. Sinusoids have big old gaps. They've got big spaces between endothelial cells. So they allow really big things to enter and leave the bloodstream. Okay, some of our biggest hormones, but then also things like plasma proteins and blood cells. Okay, plasma proteins and blood cells cannot make it through these normal fenestrated capillaries. Definitely not continuous capillaries that we have everywhere else, but the sinusoids, they can. So where do we have these sinusoidal capillaries? Liver, spleen, and bone marrow. Okay, the liver, remember, like that's where we make plasma proteins. So how else are we gonna put them in the blood if we can't fit them through somehow, right? Spleen, remember that's where we're cleaning out old blood cells. How are we gonna do that if we can't get to the blood cells? Bone marrow, that's where we make blood cells. How are we gonna put them in the blood without those big gaps present? Right, so this is all very intentional. There's reasons why we have them. Which type of capillary allows for the exchange of the respiratory gases, oxygen and CO2? Is that how you would answer that question? You would be wrong if you answered them continuous. Yes, all of them. Right? All capillaries allow for the exchange of respiratory gases. And continuous, yes, continuous capillaries do. However, they can still cross in sinusoids, fenestrated. And why? How do they get in and out? The respiratory gases are lipid soluble. So how do they get in and out of the capillaries? Diffusion, but where? Through the lipid bilayer, right? Oxygen and CO2 are lipophilic. They're lipid soluble. They don't mind that the phospholipid bilayer. They can diffuse straight through the cells. They don't need pores. They don't need gaps. They don't need channels. They don't need anything. They can just go straight through the cells. So all capillaries. That would mean continuous capillaries, yes. Sinusoids, yes. Fenestrated capillaries, yes. So that's probably an all of the above question for me for the respiratory gases. What's the definition of vasomotion? Good. So it's kind of like how we constantly change the flow through the capillary bed. And remember, that's because of precapillary sphincters, right? So contraction and relaxation of precapillary sphincters. That changes flow through capillary bed. That is completely shorthand. Conch, relax, precap sphincters, that delta flow through cap bed. You get it though, right? What are precapillary sphincters? Circular bands of muscle at what? The entrance to each capillary. Remember, when we look at a capillary is not like one nice single tube, right? We've got lots of interconnected capillaries. So like the blood comes in, and then you've got, tubes that go this way, and then they'll come and reconnect, and then split, or whatever, it goes this way, and there's all these different interconnected vessels. So going into each of them, you've got a precapillary sphincter that guards the entrance to each of these, vessels. And these precapillary sphincters contract and relax, and that alters the flow through the capillary bed, so that we make sure the flow stays, moving, and we end up feeding all the different parts of this tissue based on how much oxygen and CO2 they might need and what their pH is. What are collaterals? Good. So multiple, or you could say two, arteries leading to a capillary bed. So if that's my messy capillary bed, so if I have an arteries coming in and I go to this capillary bed, I have two arteries coming in to feed that capillary bed. That's beneficial. If something happens to this artery, say it's blocked or breaks or whatever, something happens to this artery, this tissue does not die. Why? Still another artery, right? There's still blood coming in from this one. So it's fine. It gives you kind of like a backup plan to feed that tissue. What kind of vessels are capacitance vessels? Veins are capacitance vessels. Remember, that means that they're perfectly happy holding really whatever volume of blood. It's really easy for them. If there's a lot of blood in them, great, they're fine. If you take that blood away, great, they'll be small. They don't really care. They can really easily change size without a big change in pressure. So know about the relative distribution of blood volume and the response to blood loss. So where do we have most blood volume? Most volume is in the veins, right? So by far, you've got more blood in your veins than in your arteries, your capillaries, your pulmonary circulation. Like, most of the blood is in the veins. What happens when we lose a lot of blood? So if we've got a hemorrhage, we're bleeding out, we lost a lot of blood volume, what do the veins do? Huh? Constrict. The veins constrict to redistribute that blood. Remember, the veins hold most of the blood, right? There's the majority of blood sitting in your veins or flowing through your veins. but it doesn't have to be there. We just think the veins are capacitance vessels. They don't care if they have a lot of blood or not. So if you're bleeding out, you're losing a lot of blood, you need that blood to go forward to your capillaries, right? So that you can have capillary exchange. So the veins will shrink up, you push all that blood forward, and then you can actually utilize it in your capillaries. So just in general, talking about blood flow and pressure and resistance, we kind of talk through, or we'll talk through just very, very generally speaking, like the formula for flow equals pressure over resistance. And then we'll talk through how we can change those things. And then at the end, we'll talk through like specifically how we alter blood flow, okay? And I'll make it very clear on the exam. We had like the quiz question, it was just written terribly. I'll blame someone else even though I wrote it, but it was terrible quiz question. On the exam, the questions will be very straightforward, so you know I'm talking about systemic vasoconstriction, or I'm talking about constriction of a precapillary sphincter. It will be very, very specific. So, capillary blood flow, right? How much blood is going to end up flowing through your capillaries? Flow is proportionate to, or proportional to, the change in pressure over resistance, total peripheral resistance. So how much blood flows is proportional to how much pressure is pushing it forward and how much resistance is holding it back. Okay? So know what happens if you change either pressure or resistance. So what happens to flow if I increase pressure? You increase flow. If you decrease pressure, you decrease flow, right? Pretty simple. If I stop pushing as hard, I'm going to decrease flow. If I push harder, I'm going to increase flow. Resistance. If I increase the amount of resistance holding it back, what happens to flow? You decrease flow. They go opposite, right? So what happens if I decrease resistance? Increase flow. What variables affect total peripheral resistance? So we talked about multiple different variables. We talked about turbulence. What happens if I increase turbulence? If I increase turbulence, I decrease flow, right? Because if I increase turbulence, remember that's like kind of when the blood bounces around for some reason, when it doesn't flow smooth, that increases the amount of resistance that I have, so that would decrease blood flow. We don't alter that on purpose, right? That's not something that we are, like nervous system would change turbulence to alter blood flow or anything. But there is a disease state that can affect turbulence. What disease state was that? Atherosclerosis. Okay, so what is atherosclerosis? Plaque builds up in arteries. Why does it build up? From hypercholesterolemia, or too much cholesterol. And what does that plaque buildup do to total peripheral resistance? It increases total peripheral resistance by increasing turbulence. Right? If this is my blood vessel, and the blood normally flows nice and smooth through that vessel and gets delivered, right? The blood keeps flowing through the vessels nice and smoothly. Now all of a sudden I build up this huge foamy nasty plaque all over the wall of my vessel. Now the blood doesn't flow so smooth anymore, right? The blood hits this and it bounces up, hits this, it bounces all around. You've got all this turbulence, so the blood starts just getting messy as it flows through that area. It's harder for the blood to flow. You increase the amount of resistance in the vessel, so you decrease the amount of blood that's able to flow nicely through it. So turbulence can affect resistance. Turbulence increases resistance, So decreases flow. Let me write it that way. Increases resistance, so decreases flow. How about vessel length? Right, how long the vessel is. If the vessel is longer, if you increase it in length, what do you do to resistance? You would increase resistance and decrease flow. Remember we talked about like straws, and I said, okay, imagine I have a straw that's 10 inches long, and I have a straw that's 10 feet long. Like, oh my God. Totally. That's why you shouldn't talk like that, because people are listening and making fun of you. Okay, so I have a straw that's 10 inches long, and I have a straw that's 10 feet long. Imagine drinking through the 10-inch straw. It's fine. Imagine trying to drink through a 10-foot straw. That would be really hard, right? There's more resistance. There's more area, more friction. So if you increase length, you would increase the amount of resistance and decrease the flow. Is that something that we purposely alter to alter blood flow? No, right? In adults, vessel length is pretty common, or it's pretty standard. We don't alter that. What was the third thing that we can alter to alter resistance? Diameter, right? Vessel diameter. So how does that change resistance? If we decrease diameter, what do we do to resistance? You increase resistance. If you increase the diameter, what do you do to resistance? You decrease resistance. And again, think of straws, right? Think of if you have a tiny little coffee straw, the really thin ones, or you have a nice big like milkshake straw, right? Which one is it easier to get flow through? Or which one is it easier to drink through? The big one, right? Now, again, this is just straight talking about total peripheral resistance, okay, and how that would affect total peripheral resistance. We'll talk about how we alter blood flow and blood pressure and everything in a little bit. Which of these things do we actually purposely alter to change resistance? Diameter of blood vessels, right? That's what we actually have control over. We can constrict and relax our blood vessels. Which of these things has the greatest effect on total peripheral resistance? Diameter, right? Remember, it is an exponential effect. It's like to the fourth, radius to the fourth, I believe. So an exponential effect on resistance. Diameter has a very, very big effect on resistance. We talked a lot about the velocity or speed of blood flow and how that changes throughout the systemic circuit. And then we talked about how vessel diameter and cross-sectional area relate to that. When we talked about the speed of blood flow, remember we compare that to, oh, actually I'm over on time.